Yanouri. I'm a second year PhD student at Colorado State University's Cell Molecular Biology program, and I'll be talking today about Western blots. We talked a bit about protein detection methods. We can break down these methods into two broad categories: nonspecific methods that tell us if protein is present in a sample, such as the BCA assay, or the mass of these proteins, such as a stained SES page. Or specific protein detection methods that tell us the identity of these proteins. Immunochemistry methods rely on antibodies recognizing epitopes specific to the proteins, while mass spectrometry methods rely on the fragmenting pattern and mass of the molecules detected. So let's say that you aren't convinced. You think this isn't useful. And you found this figure in an article by Mosa and colleagues looking at expression of the alpha-synuclein in different cell lines. So this is an STS page. An alpha-synuclein runs at 14.5 kilodaltons. And you think to yourself, well, this lower band right here runs around there. That must be it. That must be what I want. However, these researchers also did a Western blot and found that the alpha-synuclein was this upper band, showing that non-specific detection methods aren't quite ideal. I'd say that colleagues highlights another strength of Western blots. In this experiment, they treated male mice with ketamine for 1 hour and looked at how the subunit composition of AMPA receptors changed as a result. If we look at these subunits, we can see that they all run at the same mass. So if we were to use a non-specific method, we won't be able to distinguish between any of these subunits. However, by using antibodies with specific epitopes, we're able to check for each one and see that, yes, ketamine does lead to a change in AMPA subunit receptor composition. Through the kind of workflow for doing Western blots, first samples will be prepared and then ran across a polyacrylamide gel. The proteins will then be transferred to a membrane before being blocked and treated with primary and secondary antibodies, and then detection. Step, we're going to combine our samples with a protein buffer. If I were to do an STS page, I'd combine it with a lamelli buffer and then boil it for 10 minutes. The lamelli buffer contains beta-mercaptoethanol, which breaks disulfide bonds, as well as SES, which coats the proteins and imparts a negative charge. Boiling helps denature the proteins. We're going to load it onto a polyacrylamide gel. The percent of acrylamide your gel is made of depends on the protein of interest. The lighter your protein is, the higher percentage gel you'd want to use. You can buy them or hand cast them. I always hand cast them just because it's cheaper that way, but I did not know that acrylamide was a neurotoxin, so I might start buying them. Help impart a negative charge to our proteins. So after we load it into the gel and put it in our apparatus, a difference in charge helps pull the proteins down a electrical gradient and separate them out based on their size, with higher molecular weight proteins near the top and lower ones near the bottom. We similarly use a difference in charge to drive the proteins out of the gel and onto our blotting membrane. When making your transfer sandwich, the anode should be closest to the gel and the cathode should be closest to the blotting membrane. We want to block our membrane to prevent non-specific interactions, and after blotting it, we're going to incubate overnight with our primary antibody. Our primary antibody is specific for a protein of interest. After incubating with the primary, we do a few washes and incubate with our secondary antibody specific to the primary. So if our primary antibody was mouse anti-actin, our secondary must be anti-mouse to allow for detection of the primary. There are ways that we can detect if our secondary has bound to our primary antibody, or detect presence of secondary, either a colorimetric system, which looks for color change, chemiluminescence, which looks for the emission of light after adding a chemiluminescent substrate, or fluorescence, which looks for the emission of light in specific wavelengths after excitation with laser light. a big advantage of fluorescence is that it allows for multiplexing. If we recall the figure before talking about AMPA receptor subunits, every one of those Westerns, when they were looking at the beta-actin standard, they had to strip it and then reprobe it for whatever antibody like to see. In this paper published by Kelly and colleagues, they look at alpha-synuclein expression in neuroblastoma and Henrietta Lacks cells after treatment with a viral vector. And they see that, indeed, treatment of this vector did lead to an increase in alpha-synuclein expression. Western blots are quite fickle. This is an example of one I did, and we can see a lot of non-specific binding. This is after I had blocked this membrane for over a day. Western blots are quite time-consuming, and the quality of results very much depends on the quality of your antibodies. And it might be really difficult to find antibodies to the protein or whatever you're interested in. In such cases, other more analytical methods like mass spectrometry may be more appropriate. But thank you for watching.